The overall goal of this procedure is to simultaneously optically map free intra-sarcoplasmic reticulum calcium and transmembrane potential in the intact isolated Langendorff-perfused rabbit heart. This is accomplished by first excising the rabbit heart and cannulating the aorta for retrograde Langendorff perfusion. The second step is to eliminate contraction by adding an excitation-contraction uncoupler to the perfusate. Next, the perfusion is switched to a small volume recirculating loop, and a low-affinity calcium indicator is added at room temperature. The final steps are to warm the heart to 37 degrees Celsius using the large volume perfusion loop and to add the voltage-sensitive dye. Thus, during the experiment, changes in calcium and transmembrane potential are measured by exciting the heart with blue light and measuring the emitted light with high-speed CMOS cameras.
The main advantage of this technique over existing methods is that it allows investigators to directly probe sarcoplasmic reticulum, or SR for short, calcium dynamics without interference from transmembrane calcium signals.
This method can also help answer key questions related to the role of SR calcium handling in contributing to arrhythmias, because transmembrane potential is recorded simultaneously with SR calcium.
Generally, individuals new to this method will struggle because optical SR calcium signals are very low amplitude. Therefore, the proper loading of the calcium indicator is essential.
For this protocol, it is necessary to have freshly made Tyrode solution on hand. 4 liters will suffice.